And this is where we left off on the last one. We were talking about these tetrads. This image, I bring this image back in a bit larger later on in I'm talking about the image, the bottom image. I bring this back and maybe in a little bit larger form. But what you have, and you're not necessarily seeing here, is you have DNA breaks. You can see it in the homologous the homologous pair of chromosomes. You see this DNA break. So what you have is a duplicated set of blue ones and a duplicated set of red ones, and these are chromosomes. I'm just trying to make this a little straighter. They are held together in this synapse. And what happens is you have breaks in the individual chromosome, and where those breaks are, they can actually match up with so if we can just talk in blue and red, so the blue set of paired chromosomes can match up with the red set of paired chromosomes. Now, these are held together in the synapse, but this is prophase 1. And so they will line up down the middle, down the metaphase plate, but they are going to be, you have microtubules that are going to attach to them and pull them apart. And when they get pulled apart, where this crossing over took place, you actually are now going to have entirely different chromosomes. This crossing over was what we call recumbent combinations. And this happens really more often than you might think. It happens, as your book says, 1 to 3 times with each duplication of chromosomes. This crossing over takes place. So this adds to the potential for variety in what an offspring will bring to the table. Metaphase 1, the tetrads line up at the plate, one chromosome facing each pole. Microtubules then are going to attach via these kinetochores of each of the tetrad, and then pull the homologous pairs to separate poles. That's what's happening in anaphase 1. We're still in anaphase 1. Remember, this is meiosis, where you're going to have meiosis 1 and meiosis 2. The chromosomes move to each pole, guided by the spindle apparatus. Sister chromatids remain attached at the centromere as one unit as it goes toward the pole. And then what you have is telophase 1, the pinching in. You have that pinching in with regards to the cleavage and cytokinesis. We aren't done yet, so cytokinesis usually occurs. You have 2 haploid daughter cells. Now they're haploid because you have identicals. So you still have the identicals in there. They're not quite so identical anymore because that crossing over took place. Then you have no chromosomal replication occurring between the end of meiosis 1 and the beginning of meiosis 2. The chromosomes already replicated. We're just going to separate them now. And so division in meiosis 2 occurs in 4 stages: prophase 2, metaphase 2, anaphase 2, telophase 2. It's very similar to what you have in mitosis. Spindle fiber forms, then the attaches, line up at the plate, the metaphase plate, start to separate, and then you're going to separate into 4 haploid daughter cells, which are going to then become the gametes that will be able to fertilize and become the zygote. The chromatids that are lined up in metaphase 2, remember, crossing over took place, so they're no longer look like. They're actually their own new chromosomes at this point. They're no longer genetically identical. And then anaphase 2, the sister chromatids separate. Remember, they had been attached to each other. You had that cohesion that then is reversed by enzymes that actually pull them apart. They separate. Now you have two newly individual chromosomes toward the opposite poles in anaphase 2. And then telophase 2, the chromosomes arrive at the opposite poles. Nuclei reform, the chromosomes then begin decondensing, so turning into chromatid. At the end of meiosis, there are 4 daughter cells, each with a haploid set of unreplicated chromosomes. Each daughter cell is genetically different than each other and also genetically different from the parent cell because of this crossing over. Mitosis conserves the set of chromosomes that are produced and genetically identical to the parent. Meiosis reduces the number of chromosome sets from 2 to 1, from diploid to haploid, and also produces a genetically different set from the parent cells. And then the 3 events that are unique to meiosis, this does become something that you want to put in your head. You have synapse and crossing over in prophase 1. This leads to homologous chromosomes that exchange genetic information. So they have those attached, that crossing over takes place. You have some sharing of, or some exchange of genetic material at exactly the same locus, and then the metaphase plate, the paired homologous chromosomes, which are called tetrads at that time, instead of individual replicated chromosomes. And then anaphase 1, it's homologous chromosomes instead of sister chromatids that separate. These are the three differences between meiosis and mitosis. Sister chromatids, in terms of that cohesion, allows the sister chromatids of single chromosomes to stay together during meiosis 1. And this is the blow up that I said the other one has, which it's a little bit bigger, where you have these cohesions between the sister chromatids and then the homologous pair, and you have these little breaks in there that are going to be repaired, but they're going to be repaired very often with information or genetic data from the other homologous pair. And we call these repairs where the DNA is exchanged, called the chiasmata. Mutations, sometimes mutations can take place. These mutations can be useful, and they create variations or different versions of genes or alleles. This reshuffling during sexual reproduction is a form of genetic variation. And by the way, the sexual reproduction takes a lot of energy for the body for an organism to do this. One of the reasons why they think that sexual reproduction is most of the life forms reproduce via sexual reproduction. We talked about bacteria, etc., with binary fission. That tends to be a more energy conservative way to reproduce, but this particular way of reproduction and the fact that you have this genetic diversity is basically, the payoff is the fact that you do have species that are able to adapt, who have adapted to their surroundings and are able to continue to adapt to their surroundings. This reshuffling of alleles during sexual reproduction. There are 3 mechanisms that are responsible for this variation. It's the last thing we need to talk about in this chapter, and that's independent assortment of chromosomes, crossing over, which we've already talked about, and then random fertilization. So let's talk a little bit about independent assortment. You have 23 individual chromosomes in your body, so 23 pair individual chromosomes. And what you have is the capacity for any of these 23 to be the one that's in that particular daughter cell. So not so much that 23, but that 23 combination of that particular 23 getting together with the parent cell 23. So for humans, there are about 8 million possible combinations just from looking at the independent assortment of those individual 23 chromosomes, let alone those individual 23 chromosomes by the time they go through meiosis are no longer the same. So if you add independent assortment with the fact that you have this crossing over, the variety that is possible is just mind-boggling, that it gets to the point where each of us absolutely are truly unique. We are certainly descended from our parents, but we're still truly unique. And this, as I said, genetically and in terms of evolution, this is a positive payoff in the fact that we have so much variety that you have the chance as a species to have somebody who has the adaptability to withstand any kind of changes in the environment. Crossing over there produces the recumbent chromosomes, there's the term, which combine inherited DNA from each parent, and then crossing over contributes to the genetic variation by this combining DNA from parents into a single chromosome. Just adds to the possibility you're going to have an individual that is that much more unique. And then the last thing that adds genetic diversity is random fertilization, because any particular sperm can fuse with any ovum. So it's not predetermined that one sperm will specifically get together with one egg. It's all very random. Women actually make their eggs while they're in the womb. I mean, they're fetuses, but you know, you're making your eggs. Women are born with all the eggs they have, but that particular cycle, one egg is chosen to be the one that's ovulated. Men make their sperm when they become sexually mature. They make their sperm on an ongoing basis. But the chance that a particular egg is going to be the ovulated one is random out of about 200,000. And the chance that one particular sperm is going to be the sperm that fertilizes that egg is totally random. So this is this concept of random fertilization that adds to even more genetic variation. The fusion of the two gametes, each with an 8 million possible chromosome combination from an independent assortment, produces a zygote with about 70 trillion diploid combinations. And then if you add crossing over, you have even more variation. And so each zygote then, by absolute mathematical definition, is unique. And then the last thing, which we've kind of already talked about this, natural selection, one of the things, bringing Darwin back into the picture, natural selection results in the the genetic variation. However, remember, natural selection, the species that's most adapted to its environment is most likely to pass its genetic data along, and sexual reproduction allows this variation in populations, which can originate from mutations if you think of these crossing over as mutations. With regard to it. So basically, those that are able to live can pass on their genetic data. And that's what we have for this particular chapter.